online event of the Affiliation of Christian Biologists and the American Scientific Affiliation. My name is Brian Greuel, and I'm president of the ACB. We are delighted tonight to have ASA member Jeff Hardin as our guest speaker. Jeff is the Raymond E. Keller Professor of Integrative Biology at the University of Wisconsin in Madison. He received his BS degree in zoology and his BA degree in German from Michigan State University, followed by an MDiv degree at the International School of Theology. Jeff then pursued a PhD in biophysics under Ray Keller at the University of California in Berkeley. After postdoctoral work at Duke University, Jeff joined the zoology department, now called the Department of Integrative Biology, at the University of Wisconsin in 1991. Jeff's research focuses on the basic cellular mechanisms that transform the shape of the embryo during embryonic development. He's also an award-winning biology educator and lead author of the widely used text, World of the Cell, published by Pearson. As a Christian in the sciences, Jeff has extensive interests in the intersection of science and religion. He is an affiliate in the Religious Studies program at UW. He co-founded the Isthmus Society with historian Ron Numbers to promote dialogue between the sciences and religion, and co-edited the book, The Warfare Between Science and Religion, The Idea That Wouldn't Die. Nationally, he served for many years as the board chair of the Biologist Foundation and as an advisor to the Dialogue on Science, Ethics, and Religion at the American Association for the Advancement of Science. Tonight, Jeff is going to help us wrestle with how to situate the modern science of embryos and ourselves within the sweep of the biblical story. The title of his presentation is Fearfully and Wonderfully Self-Made, Ancient Christian Faith and the Modern Science of the Embryo. Jeff, thanks for joining us this evening.
Ryan, thanks for inviting me. It's nice to see a number of familiar faces. I'm looking forward to getting to know others of you that I haven't met yet. Well, it's a privilege to be here. I have to say that this is a different audience from the kinds of audiences I'm normally around here at University of Wisconsin-Madison. And it's great to be with other Christians to wrestle with some difficult topics. And I'm looking forward to learning from you much more than imparting any information to all of you. I'd like to start with a personal introduction. These are my two boys. The guy in red is my older son, John, and the guy in the foreground is my younger son, Christopher. Christopher has severe autism and cognitive disabilities, and I take him on a drive in the countryside west of Madison every Saturday. And I've listened to a lot of audiobooks as a result of that. And one of the audiobooks that I listened to fairly recently, and I was coming back to it from a layoff of decades, really, is Aldous Huxley's Brave New World. And I don't know if any of you have recently read this novel, but it is surprisingly prescient. And I think most of us, maybe we were required to read it in high school, but the notion that we can manufacture embryos, we can technologically manipulate them to suit our larger purposes it's just a remarkable predictor of some of the things that are happening right now that are being enabled by the technology of embryo manipulation. And we'll come back to that later. This idea that the embryo itself is self-made and that we are also playing a hand in making embryos in profound new ways that were not possible before is something that we have to wrestle with as Christians. Now, why should we be responding to this at all? Why shouldn't we just leave it in the hands of the experts? Well, let me suggest a few things. First of all, many people consider the 21st century as the century of biology. And since this is the Association of Christian Biologists, I don't have to defend myself with my astronomer and astrophysics colleagues who have served the Biologists Foundation. And I think that's really true. And it's very likely that many of us here or our loved ones are going to need to decide about ethically challenging therapies, and this involves embryo manipulation in some cases. And maybe the most significant reason for thinking about this is that Jesus in the Sermon on the Mount calls us to be the salt of the earth. And part of that is engaging the pressing issues that our society is facing. And that includes the embryo. And, it doesn't take long to do a little reading. This is from NPR. Maybe you know from February that the Alabama Supreme Court made a ruling on frozen embryos and considered them equivalent with children. This has profound effects on the legal system as just one example. But it seems like every week there are news stories related to our ability to manipulate embryos or to make things that approach embryo-like properties. And we'll come back to that later as well. Well, where are we headed tonight? First, I want to start with something that's maybe not so problematic, embryos and wonder. And I want to share a little bit about what drives me as a Christian in biology who studies embryos, because I think they're an opportunity for worship. And secondly, then we'll engage mostly with thinking about embryos in ourselves, understanding embryos in a Christian context as a starting point for at least discussing, if not solving the issues that lie at the intersection of technology and human identity. So that's where we're going tonight. And let's begin with wonder. I'm a developmental biologist. That's the kind of biologist who studies embryonic development for a living. It's an incredible privilege to study this fascinating process. And building an embryo is a staggering challenge. We all started off as a one-celled zygote. That's the image on the upper left here. Then lots of complicated things happen, imaginatively shown by the white arrow here. And then you get something really complicated as a result of this. This is my son, Christopher, about 35 years ago. And if you've ever held a baby in your arms, you can't but be amazed at the regularity of embryonic development that leads to these amazing creatures. And so it's the task of developmental biology is to try to understand at the biological level how this process occurs. And it's pretty remarkable. We start as a single cell. And by the time this is over, a human infant has about 5 trillion cells, many of them highly specialized, but not just specialized, they're organized and the embryo is constructed in a very precise way. That's not a new question. You may know this piece of Hebrew poetry. In fact, I quote from this on the first day of class in my embryonic development class here at University of Wisconsin. It's Psalm 139. Now, you may also know that Psalm 139 is ultimately about the fact that God is everywhere. And there's no place that we can be, including when we were embryos in the womb, where God has not been and is not there. But let me just read this familiar piece of poetry. For you, God, formed my inward parts. You wove me in my mother's womb. I'll give thanks to you, for I'm fearfully and wonderfully made. Wonderful are your works, and my soul knows it very well. My frame was not hidden from you when I was made in secret and skillfully wrought in the depths of the earth. Your eyes have seen my unformed substance, and in your book were all written the days that were ordained for me, when as yet there was not one of them. I've already talked about the overall context here. God was present when each of us was an embryo, including the psalmist. But notice the process language here. It's pretty interesting. You formed my inward parts. That word kilia is kind of maybe internal organs, literally kidneys. So the psalmist is aware that all of that is happening during embryonic development. And he uses weaving language, skillfully wrought. Rakam has this idea of weaving a tapestry. So it's a process and it follows a pattern. And the psalmist knew that before we knew anything about the modern science of the embryo. And it was also true that there was a time when I didn't resemble who I am now physically, and yet this same entity was me when I was an unformed substance. That's what the psalmist is saying. That's the word golem. I think maybe J.R.R. Tolkien had an idea of this when he named one of the characters in his Ring trilogy, Gollum. So these are not new ideas. And in fact, these millennial-old musings are the same kinds of questions that we ask today. The difference, of course, now is that we have insights into these processes at an unprecedented level of depth and complexity. And in fact, that's how I got hooked on my lifelong career. I was at Berkeley. I was not enjoying my lab rotation, my first one. I won't go into what I was doing, but it was involving something called the retinal pigment epithelium, and I hated it. I was pretty discouraged. But then I thought, okay, what courses did I enjoy as an undergraduate? And I really liked developmental biology. I wonder if there's anybody in the biophysics program who does that kind of thing. And there was one guy named Ray Keller. You may have noticed that I'm the Raymond E. Keller Professor of Integrative Biology these days. And so I went to Ray's lab and he said, well, here, I think you should study this. This is a sea urchin gastrula. And I looked down the barrel of a microscope at my first embryo and I was immediately hooked. I thought this was the most amazing thing ever. And it's really been driving me as a scientist throughout the rest of my career. But let's focus a little bit on ourselves and how we got to be where we are as biological organisms. And let's just take a whirlwind tour of some of the main watershed events that we might think about. And I take a whole semester to go through some of these processes with my undergraduates. I just posted my final grades yesterday. Praise the Lord. So, well, let's begin with fertilization. Fertilization is where it all begins for us. And it's a very contingent process. A single sperm has a 1 in 250 million chance or so of fertilizing an egg. And so that particular combination of one sperm and one oocyte is how each of us got our start. And this leads to the formation of a zygote. What you're looking at here are the two so-called pronuclei. This is a genetic material meeting. Eventually, that's all going to mix prior to the first cell division of this new life. So this stage is called the zygote. And the zygote gets its start in the fallopian tube, a piece of plumbing that leads from the ovary down to the uterine lining. And while that voyage is occurring, the embryo is dividing. That process is called cleavage. So here's a mouse embryo going from zygote to something called a blastocyst. And I'll just play this movie, and hopefully you can see that there are 2 cells, and now 4, and now 8. And then partway through the 8-cell stage, cellular glue becomes expressed on the surface of cells in a process called compaction. By the end of this movie, eventually what you see is an embryo in which there's a fluid-filled cavity right about now. And then there are some cells at about 2 o'clock in this, at the end of this movie that are very special that we'll come back to. This stage is called the blastocyst stage. So here's a still shot of a human blastocyst. And the cells at the very top we'll come back to. They're called inner cell mast cells. And that's what makes a baby eventually. The other tissues in the embryo, and there's some that come from the inner cell mass as well, are left behind. Those are called extra-embryonic tissues. But you came from an inner cell mass when you were a blastocyst. And we'll see that these cells are very flexible. Well, eventually, kind of a balloon-like membrane forms over the top of the embryo. It's called the amnion. That's what breaks when a woman says her water breaks when she's in labor and she's about to give birth. And then there's another balloon underneath. But the embryo itself is kind of a flat disc, and cells move towards the middle of this disc, and they dive inside, and they undergo complicated movements. And that process is the one that I've spent a lot of my career studying. It's called gastrulation. And there's a little line there called the primitive streak. It forms during these movements. And that's a very important structure as the head to tail structures of the embryo begin to appear. So here's some mouse embryos. They've been genetically engineered so that they express various fluorescent proteins in various tissues. I'm just going to run this movie. These are 3 different mouse embryos, but you can see that there's a line, a streak that forms all the way down the embryo from top to bottom. The top of each of these embryos is where the head is going to form, and the bottom is where the tail, the posterior part of the embryo, is going to form. And this involves incredibly complicated movements of the cells that form that line, moving towards the middle of the embryo to make that line in the first place, diving in the inside and then migrating away. And in the case of a mouse embryo, there are thousands and thousands of cells doing this simultaneously. This is complicated choreography. Well, after gastrulation, our central nervous system forms, and that's called neurulation. So each of us was a neurula. We all look surprisingly like a burrito, actually, and so that's what you see here. And this is the formation of what will become the spinal cord. Eventually, the part on the upper left will form brains, and the bottom part on the lower right will form the lower back part of the spinal cord. Lots of other things going on, too. This also involves some complicated movements. Here are 2 salamander embryos undergoing neurulation. You can see that there's a keyhole-shaped structure that closes over the top of the embryo to form something called the neural tube. Eventually, that forms the spinal cord. And the head end of these embryos is up at the top. Remarkable. Well, after this stage, the elaboration of that simple tube that was formed during neurulation forms things like the brain, as you see in this later fetus. Later, the limbs form, fascinating process. At the same time, internal organs are developing. External organs like the eyes, as you see here, are also being formed. Throughout all of this, cells are becoming specialized, but the embryo is also constructing itself, and that's why I title my talk Fearfully and Wonderfully Self-Made. This is an unfolding construction process that each of us engaged in when we were embryos. I'm going to give you one slide instead of an entire semester of developmental biology. What are the big ideas that underlie everything that we just talked about, all these amazing processes? Well, one of those is something called differentiation. How do parts become different? This involves changes in the expression of genes in cells that leads them to become specialized. But there is a concurrent process called morphogenesis that's also crucial. This is building the body, and this comes from the Greek morphe, which means form. So these 2 processes go hand in hand throughout all of the stages of embryonic development. And I want to make a case for why you should care about morphogenesis. It might just be because that's what my lab studies, admittedly. But I don't know about you, but my son John, he loved Legos. But he wouldn't keep them organized. He just put them in a big bucket. So if you dump the bucket out, you got all these parts, as you see on the left. And that includes some specialized parts. I think you can see a screen door in there. There are a bunch of other specialized parts. So the parts are specialized. They're different from one another, but you can see that a crucial thing is missing. They're not constructed. And that's what morphogenesis accomplishes. So the constructed edifice on the right reflects this process of form building. And that's just as crucial as making the specialized parts of an embryo. Now we're going to see that the embryo has the ability to self-organize in profound and I think sometimes unexpected ways. So this is the kind of thing that my lab studies. We genetically engineer little worms called Caenorhabditis elegans. They're sort of like fruit flies. We like to think they're cuter and kinder and gentler than fruit flies, but you can genetically engineer them, and you can watch them with sophisticated microscopes, which is something my lab does. So here's an example of that from my own lab. This embryo is covering itself in skin, and on the right, what you're looking at is a fluorescent version of a protein, a glowing protein that shows up at places where cells stick to one another. So this is part of the cellular glue that allows embryos to develop. If you're a specialist, this is called alpha-catenin. Stan Wallace, there will be a test later. All right, so that's the wonder of development. And we really shouldn't lose sight of how amazing embryonic development is. It really is a cause for worship because we really are fearfully and wonderfully made. But those processes involve a self-construction project of staggering complexity. Let's think a little bit about embryos in ourselves then, based on all of this. And this is my wife, Susie. And so Susie's the practical theologian in the house. And at this point in the talk, Susie's not here. We have my aunt and uncle are visiting, and she's been entertaining them. And but here's what she would say. She would say, life's a miracle. And what she means by that is, isn't life amazing? It is a gift from God. And I would agree 100%. But what she means is, don't we take some of the wonder away by dissecting how embryos develop? And I would not agree with that. We've had long discussions about this, and I would agree with Charles Kingsley, a 19th century Victorian theologian. He said it this way: we knew of old that God was so wise that he can make all things, but behold, he's so much wiser than even that he can make all things make themselves. For me, understanding the cellular processes of embryonic development helps me to more deeply appreciate the created order, this amazing world that God has made and that has creatures such as us in it. Well, let's then think about what we know about biology, and will it help us to think more deeply about who we are as human individuals? Well, one thing we can say from what I've already discussed is that we all started as a one-celled zygote. That's on the upper left. We went through cell divisions to form a blastocyst on the upper right. Then gastrulation happened. We got a neurulus stage embryo on the lower left. Organs formed, and eventually we all were born. The organism that's involved in all of these steps is the same organism. So there's a continuity of biological processes throughout. And in fact, what's occurring is that the potentialities that are inherent in the one-celled zygote are unfolding biologically throughout the 9 months of gestation that lead to a baby. So there's a continuity to development. That's something that we can affirm. And so the Ramsey colloquium said it this way: the embryo is human. Any being that's human is a human being. If it's objected that at 5 days or 15 days, the embryo doesn't look like a human being, it must be pointed out that this is precisely what a human being looks like and what each of us looked like at 5 or 15 days of development. Admittedly, most of us don't have photographs of ourselves as blastocysts on the wall, but we could, because that reflects a point in time in our own lives. That much we can affirm from the biology. But when we talk about the value and the identity of embryos, this raises a fundamental question: is biology really enough for this? And Sandra Wheeler said it this way: we cannot go looking for a moral status or value as if it were a feature we might locate under a microscope, if only we had one powerful enough. So biology can describe and do experiments to interrogate the biological processes of embryonic development. But things like value, the possibility, if there is one, of immaterial aspects of who we are as humans, those lie beyond the prominence of the biology. And that leads me to at least as a biologist, to adopt a position that I'll call the wisdom of reluctance. And this is really based on something Gil Meilander said. He's a bioethicist at Valparaiso University. He said this: if we're genuinely baffled about how best to describe the moral status of that human subject who is the unimplanted embryo, we shouldn't go forward in a way that peculiarly combines metaphysical bewilderment with practical certitude by approving even limited use for experimental purposes. This is a more elegant way of saying we should be reluctant to manipulate things we're not sure about in terms of their identity and value. Now, I've recently learned from bioethicist Stephen Napier a phrase that encapsulates this. He calls this epistemic diffidence. And what he means by this is that our level of caution in the midst of uncertainty should be dictated by the stakes if we're wrong. If we're not sure about what an embryo is, then we should be monumentally careful about intentionally destroying it. And that's the basic approach that Gill and Napier as well would adopt. Well, maybe at this point, it's worth thinking about what the Bible might have to say that impinges on our discussion of what exactly embryos are and whether we should be manipulating them. And we have to point out several things. The biblical authors did not know about blastocyst. The Bible is pre-scientific. So if we're looking for answers of that level of precision, we won't find them. We won't have that level of certainty. The Bible just doesn't really offer that. And as a result, Christians can disagree based on their interpretations of the very same set of biblical texts and all of this biological data. In fact, Christians do agree. I'm sure we don't have unanimity of opinion on this call tonight. But there maybe are some parameters that we can think about. Let's think about humans in the context of biblical worldview, and this is just skating the absolute surface of this question. Well, we've seen from Psalm 139, there's a continuity of embryo and adult, and one finds that in the book of Job as well. David is musing about himself before he's born in Psalm 51 as well. We can say, at least for the prophets, that embryos are clearly included as part of God's plan. God knows these prophets before they're born, so he knows them as individuals while they're in utero. And we know from the New Testament, my small group Bible study is in the Gospel of Luke, so it's fresh on my mind, I guess. Embryos seem to have personal identity in utero, and that seems to be true for Jesus and his relative John the Baptist in Luke chapter 1. What else can we say? Well, if we wind it all the way back to Genesis chapter 1, of course, one of the key features of human beings that makes them unique is that they are image bearers, or they are images. And we could talk a lot about what that would mean. That's maybe not the place for that. But one of the things it means clearly is that they're supposed to act as stewards of creation. And that might include being stewards of embryonic developmental processes. We could talk about that. There are also, the Bible makes it clear in the next chapter in Genesis that procreation involves one flesh relationships. And this leads to children who, in the beautiful language of Psalm 127, are begotten gifts. Blessed is the person whose quiver is full of them. One of the unique features of the Bible is, of course, that the weak deserve special protection, and there are boatloads of passages we could go to on that topic. But as Christians, we can say something even more profound, I think, and that is that the story of God the Son, the second person of the triune Godhead, shares our own story, because our story became his story. Of course, the Apostles' Creed says that Jesus was conceived by the Holy Spirit and born of the Virgin Mary. These are developmental events. A process lies between them, and Jesus experienced that as an embryo in his human identity. Lucy Shaw, the poet, said it this way in her poem, Made Flesh. After the bright beam of Annunciation fused heaven with dark earth, his searing, sharply focused light went out for a while, eclipsed in amniotic gloom. His cool immensity of splendor, his universal grace, small folded in a warm, dim, female space. He experienced all of that just as we did. Now, there are other things we can say about the incarnation, and maybe we can talk about that in the question and answer period. I'm particularly intrigued right now by some work by analytic theologian Oliver Crisp, and he wrote a book called God Incarnate, and in chapter 5, he talks about Christ and the embryo, and he has an intriguing idea there about when was there a full human that Christ's divine nature could unite with, as Orthodox Christians have affirmed throughout millennia. And he basically makes the case that, well, it seems like if it wasn't at the beginning, then we have a problem because temporarily Jesus had only a divine nature, not united with a full human nature. If a full human nature develops a little bit later, I'm intrigued by that argument. We can talk about that later. But there's a lot of theological resources that I think we could bring to bear on this discussion. We're not going to do that now. The main point I want to make is one made by Scottish theologian Thomas Torrance. Many of us have read his wonderful books on science and Christian faith. But he says this: in becoming a human being for us, he also became an embryo for the sake of all embryos. And that means that his becoming an embryo was an act of supreme love. And that's something that we should remember. Stuart Townend's wonderful song, How Deep the Father's Love for Us, talks about the Father's love being vast beyond all measure. And that's the kind of love that God has showered on us, even when we were embryos. British theologian Oliver O'Donovan said it this way: we discern persons only by love, by discovering through interaction and commitment that this human being is irreplaceable. The key thing here is that God loves us with an unfathomable love. And that's fundamentally how we relate to him. And that was true when we were weak, when we didn't have capacities that we now have. And I've thought a lot about this in my own context. I told you that I take my son Christopher, who's there on the right, on long drives in the country. He is profoundly disabled. And I've thought a lot about what ultimately defines who Christopher is. And ultimately, it's that God loves him, but I have an opportunity to exercise in a small way, a delegated sense, the kind of love that God has for all of us and has had for all of us since our inception, as I care for Christopher. Well, ultimately, I think we have a complicated balancing act as Christians. We should desire to use technology to prevent disease. That's what bioethics is called beneficence. And based on what I've said, I think we can make an argument that we should think about embryos as patients, as ends in themselves versus a means to some other end. And I would argue at all stages of development. And we have a healthy notion of what the Bible calls sin. So that means we should be wary of technological optimism because humans are limited. One of the interesting things is, of course, those limitations start in Genesis 2. That's before what theologians or many of you might call the fall. Before that happens, of course, the catastrophe of Genesis chapter 3 makes that a whole lot worse as sin enters the picture. So the problem is these things are in tension, and that's what makes all this really complicated. All right, so with all that backdrop, I'm going to talk about 3 examples to finish out our time together tonight before the Q&A. And I'm going to weave in some biology, which makes these things possible. And then based on what I've said, I'm going to suggest alternative kinds of approaches that we might use to avoid the intentional destruction of embryos. So the first of those is preimplantation genetic diagnosis. And this relies on the resiliency of the embryo. And I'm going to go way back to the end of the 19th century. This guy's name is Hans Triesch, and he was working with sea urchin embryos, which is what I did my PhD on. And he did an interesting experiment. He took early sea urchin embryos and he separated them, and then he allowed each of the separated cells from a 4-cell embryo to develop. Now, you might have expected you'll get a quarter of an embryo missing 3/4 of an embryo. That's not what happened. What happened was each of these cells made a little mini, perfectly proportioned embryo. This absolutely blew Driesch away. He figured, there's no way I'm going to be able to understand this, and this is inexplicable based on biology. And so he abandoned biology and became a philosopher. Yeah. Well, the bottom line is that many embryos show this kind of resiliency. And of course, we do. And the well known phenomenon of monozygotic twinning, or so called identical twinning, is evidence that the same kind of splitting of an early embryo can occur in humans. And you can get to beautiful little girls like these two girls. And it turns out that this splitting can occur quite late. It can occur up until immediately before gastrulation. So that's very late in development. So it's possible to form two separate embryos, and that requires cells to change their identity. They have to say, wait a minute, our conditions have changed. We're going to do something different, and we're going to figure it out together. And so that's what we mean by embryonic resilience. And that's what initially allowed this diagnostic procedure. In the early form of this procedure, as you see here, an embryo, before that cellular glue gets made on the surface of cells at the 8-cell stage, is held by a suction pipette on the left. And then on the right, you can see that a cell's being slurped out by a second pipette. And you could take that removed cell, analyze it genetically for genetic mutations, and then the remaining 7/8 of the embryo could be cultured in a dish and eventually, if introduced into the uterus of a mother, could develop, and you can get a baby out of this. But this relied on this amazing resilience of the early embryo. Now this is done in a different way. Something called the trophectoderm around the outside of the embryo is removed and genetically tested. That's normally not part of the baby, but it's made by the embryo, so it has the same genetic constitution. So this is how pre-implantation genetic diagnosis is done today. Well, that's cool, right? But is it good or bad? Well, depends. The intention here is to prevent disease most of the time. You can use it for other purposes. But the idea would be to prevent a really debilitating or horrible genetically induced disease. So one day it might be possible to use this as a prelude to treatment of embryos in utero or shortly after birth or those kinds of things. And that might very well be one way that it will be used. But right now, the main thing that happens is that if the embryo is deemed not to have the correct genetic constitution, it's intentionally destroyed. And that could be problematic. You can also use this for sex selection. This has been done quite extensively in mainland China and India, for example. Most people would consider that a very inappropriate grounds for using this technology. Well, I would argue it would be great if we could use an alternative to destroying embryos. And so one thing that you can do here, there is an alternative, and it's to sample a little nubbin of cellular material before an egg, an oocyte, gets fertilized. And this is something called polar body biopsy. So this is an unfertilized egg that you're looking at here, and see the little circular nubbin at the top. That's the first division product of the process that makes eggs called meiosis. And I guess since most of us are biologists on this call, that should be a familiar term to you that goes back to something you learned in high school. And you can infer the genetic constitution of the big cell, the egg, the oocyte, based on sampling the genotype, the genetics of the little guy up at the top. Now, there's another way that you can do this polar body biopsy after fertilization. That would lead to the same problems that we talked about on the previous slide. But this kind of technology seems to avoid some of the ethical dilemmas of destroying embryos. It's tough, though, and it's rarely practiced. Right, so that's one example, and you get the sense that the biology allows the technology, but then we have a decision to make about the appropriateness of the technology. Let's look at another one. Second example I'll talk about is so-called chimeric embryos. What are those? Let's begin by a mind-blowing experiment. Remember I said that at the 8-cell stage, mammals start making a cellular glue, which that kind of cellular glue is something my own lab studies called the cadherin complex. If you put embryos together after having removed a protein shell around each of them, you put them together, when they start making that cellular glue, they'll stick together, they'll glom together and make a single embryo. And that's in this case, three embryos, mouse embryos on the left were put together. They unite to make one embryo and then eventually makes a blastocyst. And in fact, if you put these into a recipient surrogate mother mouse, you get baby mice. And so here's a result of that experiment. If each of the mice has a different fur color, you put those embryos together, you make a single mouse. Well, different islands of tissue in that resulting mouse have different genetic constitutions, including the hair color that they make. And that's what you see on the bottom. And you can do the same thing in primates. Basically, you can do it in a mouse. Eventually, people have shown you can do it in a primate. In the case of monkeys, 2012, this was done in the cover article here in the journal Cell. It was 6 monkey embryos put together, and they make a single chimeric embryo. Whoa. Now, there's some evidence that this happens in humans. There are things called genetic mosaics, which look like they were 2 separate embryos that in the US merged together to make a single genetically complex mixed kind of individual that has distinct tissue. All of this is due to the incredible resilience of the early embryo, including human embryo. There are other ways to make chimeras. You can inject cells into that blastocyst stage that I mentioned earlier. And these are special engineered cells, and they will integrate into the inner cell mass, the part that makes the baby mouse in the case of the experiment shown here. And this technology has been used to make chimeric embryos starting from pig embryos and putting human cells into a pig blastocyst. And that result is shown here by the Carlos Ispasua Belmonte's group. They did these experiments. In this case, the red tissue is from humans. It's been put into a pig embryo. And you can see that most of the tissue is forming a single organ rudiment, but you can see little rose-colored cells off to the right in the head region of this pig embryo. The idea here is to use special pigs that have had their genes edited and introduce human cells into them so that when the pig develops, some of the tissues in the pig will be formed only by human cells. And then you could use those for organ transplants or for other purposes. So that's what was done in this particular experiment. And so the idea is you could get maybe this would be useful for dealing with organ transplant candidate shortages, which are a real problem. But I think you can see that there might be some other questions we could ask. And in fact, people have tried to make chimeric embryos between human and monkey embryos, also by the Ispasua Belmonte group. This was in 2019. Well, chimeras, good or bad? What do you think? Well, in 2005, our U.S. National Academies of Sciences said these kinds of studies could produce creatures in which the lines between human and non-human primates are blurred, a development that could threaten to undermine human dignity. Can't say I disagree with that. Especially problematic would be the case if human brain cells are involved and they get into the animal, or maybe vice versa. But for us as Christians, I think theologically, the most important problem with this is that image-bearing creatures are distinct from the rest of the created order. We shouldn't be mixing these kinds of embryos. And so we should seek alternatives. And one of those is something called induced pluripotent cells. So let's talk about them as our last example. Remember I said that this blastocyst, we were all blastocysts. The inner cell mass cells, those cells at the top there, they're what make the baby. But there's something that's also true about them. If you take them out of an embryo, they are incredibly developmentally flexible. They can make almost all of the cells of the embryo, and that's what the word pluripotent means. Jamie Thompson, who's developed the technology that makes the most common way of producing this possible. In a great piece, it's 2007, he said this: if human embryonic stem cell research does not make you at least a little bit uncomfortable, you've not thought about it enough. Jamie Thompson is a really thoughtful guy. He's thought about it a lot. Well, what's the technology here? The technology is take a human blastocyst and use some antibodies, which you may have heard of, incubate the embryo with these. They attach to the outer cells, the trophoblast cells, and they fall away. You can harvest the inner cell mass, that clump of cells at the lower left, and put them in a dish. And then you culture them in various ways, and you get these stem cells. They're very flexible, and they can be coaxed to make many different kinds of specialized cells and tissue types. But the problem here is that making human embryonic stem cells involves destroying a human embryo. Well, is that good or bad? Well, this always involves intentional destruction of embryos, and based on your view of the embryo, that could be definitely a negative. And it would be desirable for everyone concerned if there were alternatives that were available. And it looks like a really good alternative is something called induced pluripotent cells. And they're possible because of this gentleman, Nobel Prize winner Shinya Yamanaka, received a Nobel Prize in 2006. He determined which genes are expressed in these very specialized pluripotent cells using mice. And he determined that there were 4 main proteins, which are now colloquially called Yamanaka factors. And if you force any specialized cell, or most specialized cells, to make these 4 proteins, this resets the specialization of those cells so that they become pluripotent cells. And there's a way that you can harvest them. And then you can coax these induced pluripotent cells to make specialized cells, and they behave in ways that are almost indistinguishable from embryonic stem cells. The difference here is you can start with cells that are genetically matched to a patient. No embryo destruction is involved. And the idea would be that you could coax these to make differentiated cell types that human patient might be lacking, for example. Amazing. But there's also a problem. These cells are incredible. If you put these cells together and you provide various culture conditions, they will self-organize in freakishly dramatic ways. And scientists are doing this all the time now to make things that are called organoids. Organoid sounds like organ. These are organ-like structures starting with pluripotent cells. And here's an early example. This is something called a cerebral organoid, and this is from mouse cells, but you can see that it looks like a slice of a brain. And that's because these induced pluripotent stem cells have been coaxed to become specialized to make nerve cells, and they organize into a brain-like structure. And you can show that some of these brain-like structures have rudiments of the electrical activities that we can discern in our own cerebral cortex, for example. That's interesting. We're not going to talk about that particular example. I'd like to talk about another example. It's possible to take several different cell types, some of them derived from pluripotent stem cells, and mix them together in specialized kind of microengineered wells, and they will self-organize into things that look remarkably like actual embryos. And I'll call these synthetic embryos. They've gone by a number of different names, but a number of groups are using this technology. The embryo on the left is a normal mouse embryo. The entity or the biological conglomeration on the right is made from induced pluripotent stem cells. This is an embryo essentially arising from a dish. Now, it doesn't have all of the properties of an actual embryo on the right, but it passes the smell test to most people who are not used to looking at advanced mouse fetuses. And this technology is moving at an unbelievably fast pace. And I was just at Harvard, recently heard a talk by Jakob Hanna from the Weizmann Institute in Israel. And he and a number of other groups, one of which I'll mention momentarily, are really pushing this technology very rapidly. And they're combining it with an ability to culture embryos ex utero, so in a bottle, essentially. And these two technologies in the next couple of years are going to progress very, very rapidly. And so this is creating some controversy. So this is Magdalena Zemeckis-Getz. She's at Cambridge. She's at Caltech now, but she's initiated a lot of these experiments when she was still at Cambridge. And she and Jakob Hanne's groups have been able to make embryos that look remarkably like human blastocysts, starting with induced pluripotent stem cells or embryonic, human embryonic stem cells. And the question is, what exactly are these? And as I say right now, they don't develop in ways that indicate that they have the properties that normal embryos have. But I just remember I said that the technology is going to move quickly. In a couple of years, remember I said that tonight. Now, when this technology was just beginning, a group at University of Michigan who was making these synthetic embryos looked at them and said, wow, they sure look like actual embryos. And at the time, one of the investigators there said, we have to be careful using the term synthetic human embryo because some people are not happy about it. Yeah, no kidding. We have lots of ethical questions we're going to have to address here. Why? Well, people are trying to take synthetic embryos in the case of monkeys and implant them into surrogate mother monkeys. They've tried this. It has not succeeded so far, but Jakob Hanne's group and others are working hard to try to get this to go. In Jakob's case, he's using these technologies for other purposes as well, mainly to try to develop human organ cultures, also to try to offset the human organ shortage. Well, those are just 3 examples of how amazing embryos are. And because of the very properties biologically that embryos have, it allows technologies that in some cases are exhilarating, in other cases might be disturbing. And so what are the next steps for all of us? Well, I think number 1, be informed. It's tough to stay on top of this. It's moving really quickly. I hope I've helped a little bit tonight. Be critical thinkers. Don't believe everything you read. Don't believe the claims that some people make. Be nuanced in your intake and evaluation of what you read about these things. And we're going to have to be loving advocates. Remember, Jesus said, be salt of the earth. We don't want to rub salt in wounds. We want to be a preservative function in the wider culture. And that means we need to be profoundly Christian, and that means being loving entrance into these kinds of discussions. That's all I have for now. I'm hoping we'll have some interesting discussion in the Q&A. Thanks for your attention. It's great to be with you.
Thank you, Jeff, for that informative and thought-provoking presentation. We're going to start with 2 questions that were submitted in advance by Luke Jansen.
Jeff, thank you very much for that talk. That was an amazing walk through embryology. I really appreciated that. And I also am glad that you talked about the twinning process, how one embryo becomes 2 or 3 in case of triplets. And you also talked about the chimera, where at least 2 different embryos can come together and produce one being. And so then my question has to do more with the theological implications of this and the whole idea of the soul. Have the theologians stepped in and talked about whether in the case of twinning or triplets being formed from the one embryo, do they have multiple, would they each have their own souls? And I think many would answer very quickly they would. But then if you look at the chimera, where you have 2 or 3 very different individuals with different genotypes being put together into one being, does that person then have 2 or 3 different souls?
Yeah, I mean, these are great questions. You'll notice that I didn't really adjudicate on the question of soul, and I guess we could talk about a little bit about what ways that people thought about that. Now, Truth in Advertising, Luke has actually written a book on the notion of the soul called Soul Searching. Love the title. And I think different Christians have different understandings of whether there's an immaterial aspect to each of us, which would complicate the interpretation of the biological experiments, for sure. I think the Old Testament tends to, without getting into the weeds, which I'd like to avoid, but the notion of nephesh, maybe it's better to say that we are souls than that the Old Testament doesn't seem to think that we have a material essence which is separate from our physical essence. We're kind of psycho-spiritual, physico-unities. I think that tends to be how the Old Testament portrays things. And but in the early Christian period, as Luke well knows, Greek philosophy was in dialogue with early Christian thought, and from there we get some of our modern notions of the soul, and that's all. I think it's hard to know how we should think about some of those questions. I guess what I'll say is, there was a early medieval, kind of the beginning of the medieval period, theologian Bertius, who defined human persons as an individual substance of a rational nature. And you just have to unpack all of those ideas, all those words. It's complicated. That might be a valuable definition to this day, but I think most of us think of one of two forms of what the philosophers and Stan and maybe other philosophers on the call can weigh in here, what are called substance dualism. So the modern form of that would be Cartesian dualism. So we have a body and we have a soul, and it tends to favor that our identity is mostly wrapped up in our soulishness and not so much in our body. But then St. Thomas Aquinas, kind of via Aristotle and Bertius both, developed a notion of, to use a big word, hylomorphism, or what's often called Thomistic substance dualism. And that's more complicated. So there's an organizing principle that is part of a human organism, and sometimes it seems like it's an immaterial essence, but other people almost talk about it as if it's a systems level understanding of the biological parts, and that's pretty complicated. All of this is to say that Luke's right. This makes it complicated to think about particular experiments, and I just got a couple of things to think about. All right, so here's our lovely girls again, right? My own view is that in the case of twinning, that's maybe easier to think about, at least biologically. Some people make the argument, it's only until after twinning is no longer possible that you have an individuated human being. And my problem with that is that no one would say that we had, when they stop to think about it, there's never a time when there were 0 human beings and now you have 2. You had 1 and you get 2 for the price of 1. And so there are other organisms where this happens all the time through asexual reproduction. So I have a colleague, Phil Newmark, he studies planarians. You can cut flatworms and they'll regenerate. One flatworm cut in half will regenerate to form 2 worms. Now, it's clearly there's one worm before. The splitting process kicks in some biological activity, which results in 2 organisms. So at least biologically, that's what I would say is happening in twinning. And maybe it's injury or maybe less than full adhesion early on or something, but there's some biological impetus that leads to the splitting of the embryo, and then the self-organization of both of those leads to 2 individuals. I think there are ways that substance dualists would explain twinning, that God infuses a second, into one of the embryos, maybe has the first immaterial essence, and then God, if you're a creationist, which is another issue about where souls come from, or we could talk about that. But I think there are ways to understand that one. Now, the chimeric embryo one, I don't know what happens with immaterial essences in that case, but I do have a sci-fi analogy for you. So, my son John and I watched Star Trek Voyager. Maybe some of you did. I'm not sure whether that was how great that was, but there was an episode there where there was a guy named Neelix. He's the guy on the left, interesting looking fellow. And then there was this Vulcan guy named Tuvok in the middle. And they were in a transporter accident. And so they were combined to make a single individual who was named Tuvix. Now, I think Tuvix was his own person. He was a unique individual that resulted from the combination of these two pre-existing individuals. So at least biologically, it's probably how to think about chimeric embryos. I think chimeric embryos, there are ways of thinking about that. Some people might propose God takes away one immaterial essence and leaves one remaining, but you would have to invoke some sort of more complicated explanation if you're a Cartesian dualist, for example. So I don't know if you have thoughts on that, Luke.
So I do have thoughts, and you did articulate essentially what my thought would be, that there's difficulties if we hold the view of substance dualism, two different things, a soul riding in a body, and the bigger problem, I think, again, is with that chimera. It does happen. By the way, how often does the chimera situation come up in real life?
Is Earl Godfrey on this still on the call? He actually is a human embryologist. So he might know the answer to this. It's very rare, but it's often not detected because you're not looking for it. So I don't think we have good stats on that. I think it's quite rare, though.
I am not a human embryologist, but I teach them, people who do IVF.
So do you have a sense for how prevalent human chimeras are, like naturally occurring human chimeras? I don't either.
No, I don't. I haven't heard anything about it.
Okay. Stan Wallace, you want to unmute yourself and ask your question?
Sure, Jeff. Again, so appreciated your comments. I'm very, very interested in this whole issue. I feel like a philosopher among the biologists here, but interested in the same range of issues. And I was interested in your comments about Aristotelian hylomorphism and how that relates to this issue, because so much of what you said from where I sit was a hylomorphic vision, right? That there is the nature, the essence of the being, the person, that's constructing the body in this teleological way toward a certain end that's very clear in terms of the developmental pathway. And of course, Aquinas then, I think, integrated that with Christian thought in a very helpful way. I am wondering if that's much of a conversation among developmental biologists these days, as there's more work being done, I think, in the Aristotelian hylomorphist frame of reference, and if so, how that's being received by the broader biological community. And while I'm at it, I will put a link in to a helpful treatment by a philosopher on cloning issues. So I'm going to stick that in the chat while you're responding. So thanks.
Well, sorry, Luke, I'm going to jump in over top of you. Is that okay? I'm going to go answer Stan here. So, I have to say, in the academic guild of biologists, Aristotelian approaches are not it's hard for Aristotelian voices to be heard. I would say that. Now, in defense of that, I was part of a grant program through the John Templeton Foundation where I really wanted Aristotelian thinkers to be part of discussions about the self-organizing properties of embryos specifically. So, I've talked to a number of them, and at times the language, as I said, shades almost into systems biology. I think they're not of one voice about how they think about these things. But yeah, I think a very articulate couple of hylomorphists, their brother and sister. I went to grad school with the sister, that's Maureen Condick, and her brother Sam Condick. They're Roman Catholic. It's a very prevalent position among Thomist philosophers and theologians in Roman Catholicism. They've done a good job of trying to talk about some of these key issues. Maureen wrote a book on twinning, for example, and Maureen and Sam wrote a book on, which basically takes this Aristotelian or Thomistic really position on embryos. So that's one thing I would say. And thanks for the link in the chat.
Luke, did you still have a question related to the talk?
Well, if there's not other questions, that's actually a remnant from our previous conversation. I was reacting, and then so I guess maybe I'll do one quick follow-up.
Okay.
The one point that does come up with what was said earlier was that any interpretation of the soul that involves substance dualism of any kind, I think it becomes problematic because of the things that we were talking about earlier. And the problem is that Christianity, I think, and many people think Christianity insists on substance dualism, although there are Christians such as myself who don't hold to that idea. But here's a follow-up question then. Again, with this idea of a chimera that involves what it seems to be two or sometimes more individuals in one blastocyst, in one person, I wonder if this comes up in conversations and discussions amongst people about the whole trans, sexual issue, where people say, I feel like the other gender. I know I look like this, but I feel like the other. Do people talk about this related to being a chimera situation again? I'm not sure what the terminology to use, but there's my question.
Yeah, I mean, I guess I feel like those topics are beyond the scope of what we want to do tonight. Happy to maybe have that discussion offline. Feelings that requires embryos that are quite advanced, organisms that are advanced, and psychological. There's a psychological component, physiological, all kinds of things there. Maybe I feel like that might get us off topic on from this particular discussion. There have been some claims of genetic mosaics where part of the mosaic is XY. That would be a genetically male individual and XX cells. It's possible to explain how those arise. But I think we just don't have enough information about those, in my view, to really say very much about them. I would say in defense of substance dualism, there are things that substance dualism does a pretty good job of that are problematic for certain other views. So, and one of those is personal identity through time. And, there's some other issues about resurrected bodies and other things that would take us way off topic. I'm a biologist, and so I'm very interested in thinking about how the biology interfaces with philosophy and with philosophical theology. My sense is that my secular colleagues at Wisconsin, maybe Luke, you might feel this way too, feel that some substance dualist arguments seem ad hoc. And I understand that, but there are other reasons for being a substance dualist that involve more fully mature humans as well, and thinking about them not just before birth, but after death and everything in between. And so I would want to be careful that while any kind of metaphysical position has certain problem areas that it has to deal with. the other metaphysical positions have their own problem areas. And we probably don't have time to go through all the pros and cons of all of those here, but I just wanted to make that particular point.
First of all, thanks so much for your talk. Really appreciate it. As someone who trained as a developmental biologist and still teaches developmental biology every fall to undergraduate students, and who also teaches bioethics, these are things that my students wrestle with quite regularly because I'm at a Christian institution. And what I think they are most challenged by, and I wanted to get your perspective on the difference in teaching in a secular environment, some of these same topics, is how God's providence just shines through in these mechanisms, and the fact that, in him and through him, all things are created and sustained really resonates with my seniors when they move from a developmental biology class in the fall and then into the bioethics class in the spring, where we talk about subjects like pre-implantation genetic diagnosis and in vitro fertilization and use of human embryonic stem cells. But we're able to unpack that in the context of having a Christian worldview and a biblical foundation and understanding that God is providential. So with your experience teaching developmental biology and dealing with some of these issues, how is that different at a secular institution? Are students more challenged by that, or do you think they're less challenged by that? And again, appreciate your time tonight.
Could I just ask a clarifying question about the last part of that? You're asking whether my students at Wisconsin, they are more troubled by the ethical dilemmas or less troubled by them?
Yeah, essentially, how do they deal with them without that certainty in Christ that the rest of us here tonight have?
Yeah, that is an excellent question. I would say some of the students have never thought about any of the ethical underpinnings for anything that they believe. So I had a student number of years ago. So I do, it's a biology class, but I spend about 2.5 lectures worth on talking about ethical issues. And I just talked about worldviews and talked about, the ontology of embryos and where do our ethical systems come from, and like what's the support for how we think about what the nature of the good is, and what is the nature of humans. And, we can't unpack the word person very much, but that kind of thing. But, I had a student who came in and she said, after your lecture, I realized I have no earthly idea where my ethical conceptions come from. And we ended up having a book study of Mere Christianity, and she ended up becoming a Christian. And so there are those kinds of students, but I would say she's not typical. So the typical kind of student just says, well, we want to help people, don't we? And so they have a very kind of naive view, which is sort of, non-Millsian utilitarianism, basically, about combined with an intuitionism about the early embryo. Well, it just doesn't look like it deserves the kind of same moral respect that more fully developed humans deserve, that kind of thing. Now, there are some students who are very articulate and, they'll talk about higher brain function being a watershed or some other point in development. But I would say my goal is just to try to get them to realize that they haven't thought about this enough. Jamie Thompson's quote, I give them that quote because he is not a Christian, but he's super thoughtful. And, but I definitely open the door to them to say, our religious commitments inform our worldview. And I identify as a Christian and say that clearly informs my worldview. I talk about my son Christopher, that his name means Christ bearer. Well, he's God's gift to us. We never thought that he would be the bearer of Christ to us and that we would feel Christ's presence more deeply by having a severely disabled son. But I would say, women's bodily autonomy is just a huge kind of given for many of my students, and they think that obviates all of the ethical dilemmas. Basically, well, it's an individual woman's right to make decisions about anything inside of her body, and that's that. But I do try to make the case that, well, no, it's a lot more. It's more complex than that. We can talk about that, but. Yeah, I wonder about your students. How do they? So maybe they have something they've been taught that they should think about all of these topics when they come to you, and then you're helping them to go deeper and think more deeply about these things. What are the special challenges you found?
By the time they take the capstone bioethics class that I teach, they should have had 4 years of rigorous scientific education, a solid foundation in Old and New Testaments, intro to theology, worldviews, comparative religions, philosophy. So they should be thinkers. And so when I introduce my class to them, I often challenge them with this. I tell them, I don't care what you think. I care that you think. If your only justification for X, be it abortion or PGD or IVF, whatever you want to throw out there, is because grandma told me, or in 7th grade, I heard my youth pastor say it, we're going to have a problem. I need you to understand the topic thoroughly, form an opinion, and then be able to defend that opinion. And I make them do things like they do mini debates in classes, and I make them defend both sides. And I won't tell them until they're standing in front of the classroom whether or not they're pro or con of a subject like pre-implantation genetic diagnosis. Because I tell them, you can't have a winsome conversation with someone if you have no idea what their position is or where they come from. And at first they can be resistant to that, but by the end of the semester, they just embrace it because they've finally taken sort of that last step in becoming mature thinkers and applying their biblical knowledge and their Christian worldview and really having this robust understanding. Oftentimes, I will get the problem, though, that they say, I thought I knew what I thought until I took your class, Dr. Parker, and now I don't know what I think. But I do think about it, and I think about it well and deeply. So it's an interesting dichotomy that oftentimes they come in similar to your students. Well, I just kind of have this intuition that this is the way it should be. And then I try to help them, think through that maturation process intellectually. But it's quite a fun topic to take them through.
I missed the first several minutes of the lecture, so you may have touched on the issue of abortion, but it seems to me like this is a kind of the hidden agenda from the lecture. Do you feel that this is a legitimate topic for evangelical debate, or is it a no-brainer? For example, from your presentation, I suspect that you would be against all abortions from the fertilized egg onward. So could you comment on your position on that?
Well, I don't think there was a hidden agenda in tonight's lecture, really. I was just trying to explain how I think about these things as a developmental biologist and from an understanding of Scripture. I would certainly myself seek to minimize the number of abortions that occur if we could do that. I think that's a good. So I definitely would say that. Um, phrases like consistently pro-life maybe would characterize my own view. I think we should care for people who are pregnant. We should care for the developing life within them. We should care for babies after they come into the world. We should, for me, that's all part of a total package of being a loving Christ follower. There may be times when abortion is necessary to save the life of the mother, for example, and we can talk about those kinds of things, I guess, where to me, most of the situations where that's invoked maybe don't. I think that situation is rarer than maybe sometimes it's portrayed to be. But yeah, I mean, certainly my position would be conservative on and destroying any unborn life.
Let me give an example here. If you take a person who is 3 years old and he's murdered, and there's a 30-year-old person and he is murdered. The legal implications of the difference in ages are non-existent. They would both receive the same sort of treatment. However, in the development of the embryo, if you take a 3-week-old embryo and compare it to a 30-week-old fetus, even the most extreme anti-evolutionists would see that as a different value. And if they deny that, then why is it that they always throw up their hands and say he agrees, he supports abortion, even just before birth? It's clear that they also, whether they would admit it or not, would see a different value in the life of a 3-week-old embryo and a 30-week-old fetus.
I'm not sure everyone would see that difference in the among the group that you're talking about, but I do think we have to be careful about appearances. Appearances are. So if we, as I do, believe that God used evolutionary processes to bring us about, we don't have evolutionary equipment that helps us think well about early embryos because they're just not part of our evolutionary history in terms of no obvious survival value there, maybe. And so we do have to be careful about how we feel about embryos at different stages just by looking at them. I think it's a very common sentiment among my colleagues. Well, it's just a clump of cells, for example. You'll hear something like that. Well, not really. It's a self-organizing individual human organism. Not all of its potentialities have been actuated yet, but given enough time, it's going to do that. So I think it's maybe a little more complicated than I hope it's more complicated than you're laying out for some of the people that you're describing.
Yeah, well, it's great to be with you, and thanks, Jeff, for your presentation. This is a time to have a conversation with one another as well. And I just have a very quick comment on the comment that was just raised. It seems to me, first of all, there are a lot of people who don't recognize the difference between 3 weeks and 30 weeks. But secondly, of those that do, I don't know that it is always or even mostly because there's a difference in value attributed as opposed to a different epistemic clarity. There are a lot of people who are just more certain of the value, but not that there's greater value.
That's probably right, Jeff. That gets back to the Stephen Napier idea of epistemic diffidence. Like if you're not, people have more confidence. And I'm sorry, I have already forgotten the name of the person who posed the original question. I'm so sorry. But if that someone might not have the same epistemic confidence about early embryos,
Right.
And would assign the same value at all stages of development. Yeah, I totally agree with
that. Jeff, this will be a quick and easy one, I hope. We often hear this, the passage that you read, Psalm 139. People often will refer to that passage, how we were formed in our mother's womb. In the first half of that passage, it says about being formed in the mother's womb, but then we often don't look at the second half of that passage, which talks about being skillfully wrought in the depths of the earth. Just curious what you might have to say about that.
Man, I've asked several Old Testament scholars about this. The easy answer is, it's a metaphor, right? Other people have more complicated interpretations of that based on ancient Near Eastern literature and how things are, embryos are formed from mud and, other kinds of contexts. I guess I normally think about that as the womb is dark, the underground is dark, there's absence of light, and so that's essentially a metaphorical description. Now, you might have a different take on that, but I haven't. Yeah, I'm not. Some of the more convoluted theories, I guess I'm not convinced of them, but maybe you have a different take on that.
Well, I don't know if we have time, but I would be one of those that does. I wonder if the ancient Hebrew writers borrowed from some Babylonian ideas there, but maybe we can talk about that another time.
Yeah, no, definitely. They're swimming in the same intellectual milieu. It's in detail, what did they mean by using that phrase in Psalm 139, maybe is the question. Just like we know that they were influenced by the Gilgamesh epic and all of the other, all of the other ancient Near Eastern creation epics, you can see the reverberations in Genesis, but just how one should think about that in terms of your hermeneutics of the biblical text. That's the question that I have, I think. So, I'd be love to continue the dialogue. I also need to read your book carefully. So I have it and I've started it, but I haven't finished it. So maybe after I read it, you and I can chat. Great.
Well, lots of good questions. And clearly there's an interest in this topic. So maybe we'll have to have a follow-up lecture by someone that relates to this. Thank you again, Jeff, for your wonderful talk. Let's close out the evening in prayer, and I'd like to ask Carl Pinkham, our Vice President of the ACB, to lead us in prayer.
Thank you, Brian. Let's pray. Lord God Almighty, Creator of the universe, we thank you for the wonder and precision of your plan to bring us from two joined germ cells through the myriad of precisely orchestrated cell divisions, dances, and dialogues to a child about to be born. We thank you for Jeff Hardin, who gave us knowledge this evening to help us understand this process, to help us know better how to protect and respect the unborn and the mother carrying the unborn. Thank you for his clear message that our current knowledge of human development and ways to manipulate it brings us to places where we must rely upon you for the wisdom to choose what is the right and loving way to respect human life. Bless all those who participated in this event tonight. Bless the American Scientific Affiliation, and bless the affiliation of Christian biologists. We pray all this in the name of the Father, the Son, and the Holy Spirit, the three-in-one God. Amen.
Amen. Well, good night, everyone, and thank you again for joining us this evening. Stay tuned for future sessions of the Affiliation of Christian Biologists. We'll put out the word when the next one is. All right, good night.